Welcome to tonight's Camaraderie. My name's Terry Humphreys. Welcome to the last edition of this year. It's been a good 8 of them, as I said earlier, so thank you all for coming. I'd like to acknowledge the traditional custodians of the land we are meeting on, the Whadjuk Noongar people, and I wish to acknowledge and respect their continuing culture and contribution they make to the life of this city and this region. I'd also like to thank the sponsors of this year's series, the Chem Centre. It's been fantastic having them involved this year, and our bellies have been very happy at the end of every event. So we'll move on to the main event, which is the Camaraderie talk by Associate Professor Matt Piggott from UWA, and he's going to be talking about drug discovery in the Piggott Group. He's got quite the history and awards and things like that. He's mainly teaching and research academic medicinal chemist at UWA. He graduated from UWA with majors in chemistry and biochemistry and molecular biology. And then he went on to do a PhD in natural product total synthesis under Dieter Vega in 2000 at UWA. Then moved on to his first postdoc with Martin Banwell and Chris Easton at ANU. And then he worked with T. Ross Kelly at Boston College towards a chemically powered molecular motor before returning to ANU. So he's got extensive experience in drug discovery and preclinical development for cancer, trypanosomosis, neuropsychiatric conditions. As you can tell, I'm not a biologist. Parkinson's disease and other neurodegenerative disorders. He is co-founder and director of Cetonix Pharmaceuticals and startup spun out from UWA and the Telephone Kids Institute, and he's also sponsored collaborations with local and international biotech companies such as Imria, Neurovigor, and has previously collaborated and consulted with GSK, EBU Pharmaceuticals, Atuka, and a few others. So after that, thank you very much for coming along, and please welcome Matt to the stage.
Hi everyone. I just want to start by thanking Terry and Keely for the invitation to speak tonight, and Chem Centre for their ongoing sponsorship of these events. Terry and Keely do a great job of camaraderie, and I think we all appreciate their efforts. So as Terry mentioned, my training was in synthetic organic chemistry and the total synthesis of natural products, and that's still one of my favourite research activities, but it's kind of like an expensive hobby because I guess I'm not good enough at it to get funded for that kind of research. So in my independent academic career, I've moved over to medicinal chemistry, and to various extents, all the projects you can see up on the board here involve some kind of collaboration with industry. So we've dabbled in drug discovery for trypanosomosis, because the disease is caused by parasites that cause African sleeping sickness and Chagas disease. And we did some work with GlaxoSmithKline through the Tres Cantos Open Lab Foundation on that project. Collaborate with Neurovigor Therapeutics, they're a startup company coming out of Purdue University in the States on reactive aldehyde trappers. Reactive aldehydes are implicated in a lot of different diseases. And as Terry mentioned, we spun out a company, Cetonix Pharmaceuticals. My co-founder is Joost Lesterhaus at the Kids Institute, no longer the Telephone Kids Institute, just the kids now. And that is looking to discover targeted treatments for cancer, and the target in that case is a protein called p-pi gamma. Obviously I can't talk about all of those things tonight. The title of my talk is a bit misleading. I'm going to focus on this one in the middle here, and that is drug discovery inspired by the illicit drug MDMA. Well, it was an illicit drug, it's not anymore. And this has been bolstered recently by a major collaboration with a local biotech company, a clinical stage biotech company, Emiria Limited. And I'd like to join the other speakers in an acknowledgement of the country. We live and work on Whadjuk Noongar Budja, and we're currently in the beautiful second spring season in the Noongar tongue, that's Camaraderies. And any kid who grew up in the southwest of Western Australia knows that this is the season where you've got to look out for swooping Kulbadi and Buttala. So MDMA is the inspiration or the lead compound for therapeutic drug discovery in my group. And of course, it's better known as a recreational drug. It goes by the street names ecstasy and molly and a bunch of others. So MDMA was rediscovered by Alexander Shulgin in the late 1970s. And Shulgin was a maverick medicinal chemist who made hundreds of different psychoactive compounds and tested them on himself, sometimes with his friends. And he documented his observations. Been really valuable research for our research. One of the things he said about MDMA in his famous book, Pical, is that it induces an easily controlled altered state of consciousness with emotional and sensual overtones. It has been described as a psychedelic, but it's quite different from the classical psychedelics like mescaline, which causes hallucinations. MDMA doesn't really cause hallucinations. Instead, it's the prototypical member of a distinct class of drugs called the entactogens. That's derived from Latin words which literally mean producing, touching within. So it's a drug that allows people to get in touch with their inner selves. It also promotes feelings of disinhibition, trust in and connection with others. And these properties make MDMA ideal for psychotherapy. And Shulgin and David Nichols, his collaborator, got in touch or spoke about MDMA to some of their friends and colleagues and recommended that MDMA might be useful for psychotherapy back in the late 70s, early 80s. And it was used for that purpose, and quite successfully apparently. But at the time, MDMA was also widely used recreationally, and the United States government didn't like that. And so in 1986, it was rescheduled as a prohibited substance. And that, of course, curtailed any further use in therapy and research in the area for many decades to come. If you want to learn more about the history and chemistry of MDMA, I recommend these reviews. So, fast forward about 30 years after much campaigning, Rick Doblin and the Multidisciplinary Association for Psychedelic Studies, after a lot of work, finally managed to get MDMA into clinical trials for the treatment of post-traumatic stress disorder. 23 was the first readout of the phase 3 clinical trial, and it was hugely successful. About twice as many patients who had MDMA along with the psychotherapy compared to the patients who had placebo along with the psychotherapy had benefit. And by benefit I mean essentially a cure. They no longer met the diagnostic criteria for PTSD. So twice the success rate with the drug as opposed to without it. On the back of those amazing results, in June of 23, and quite a shock to the world, to me it was a shock to me anyway, Australian TGA rescheduled MDMA for the treatment of post-traumatic stress disorder. So it went from Schedule 9, a prohibited substance, to Schedule 8, a controlled drug. And that was essentially a world first. At the same time, they rescheduled psilocybin, which is the psychoactive component of magic mushrooms, for treatment-resistant depression. And Ameria were right onto this in January 24. They applied for and received approval from the TGA to administer MDMA-assisted psychotherapy for PTSD, and they started treating patients soon after. In the middle of this year, the US FDA rejected a bid from Lycos, which spun out of MAPS, for similar therapy, MDMA-assisted psychotherapy in PTSD. And you might think that that was a bit of a blow for Ameria. It would have been nice if the FDA validated what Ameria is doing here in Perth. But on the flip side, you would think that any future application to the FDA is probably going to rely heavily on the data that's coming out of Ameria at the moment. So they have potential to really influence things going forward. The other thing is, as far as I'm aware, Ameria is the only company in the world that's delivering MDMA-assisted therapy in a kind of scalable way. So there's a potential for kind of mental health tourism, people to come to Western Australia, to Perth, to have this kind of therapy. So things are going well. They've treated 16 patients. That involves about 40 drug sessions where patients are getting the MDMA as well as therapy. And about 50% of the patients are in remission, no longer meeting the diagnostic criteria for PTSD, and all of the other patients have had clinically significant benefit from that therapy. And Emeria are branching out into other psychoactive substances, treatment of mental health conditions. We're about to initiate a methylone trial. So methylone is much like MDMA, it has this extra ketone carbonyl group here. That makes it a cathinone. And this is a bit different from the MDMA-assisted psychotherapy in that it'll be a drug-only trial, so no therapy along with it. So it'll be interesting to see how that contrasts with MDMA-AT. And they're also working towards an application to deliver psilocybin assisted therapy for treatment-resistant depression. So that's on the cards for 2025. And they're working hard and undergoing negotiations with the payers. At the moment, MDMA-assisted psychotherapy is very expensive. It costs between $25,000 and $30,000 for a complete regimen of treatment. And obviously most people can't afford that. But they're talking with the Department of Veterans Affairs, obviously very relevant when it comes to PTSD, and two private health insurers. Negotiations are going well. Once those payers come on board, it will make the therapy available for a lot more people. So MDMA seems like it could be a bit of a game changer when it comes to PTSD and maybe other types of mental health therapy. But it's not a perfect drug. There's no such thing as a perfect drug. For assisted psychotherapy, its half-life is too long. This means that clinical sessions are long, they're about 8 hours, and that makes it quite expensive, as I mentioned. It's also a stimulant, just like having too many cups of coffee. It causes anxiety when it's first kind of kicking in, and at lower doses, and it also increases blood pressure and heart rate, and obviously that's not conducive to psychotherapy where you want the patient to be relaxed. It's also not applicable to people who have cardiovascular problems. It also causes intense euphoria, and that is an abuse liability. It remains to be established whether that euphoria is important for its therapeutic benefits, but that's the kind of research that people around the world are doing, including us. Those problems, you can get around those problems in the setting of assisted psychotherapy, where there might be 1 or 2 sessions and they don't last for too long. But if you want to use an MDMA analogue for a longer term therapy, where you might be wanting to take a drug every day, MDMA, and especially its metabolite, which is methylene dioxyamphetamine, where this N-methyl group has been metabolically cleaved off. They're both agonists, so they activate the serotonin 2B receptor. 5-HT stands for 5-hydroxytryptamine. It's another name for serotonin, which is a neurotransmitter. And drugs that have this kind of activity cause heart valvulopathy, a type of disease which can lead to heart failure. People have died from this kind of disease, and several drugs have been withdrawn from the market because they have this kind of activity. The other thing is, no one, or at least most people, wants to be high all the time. So if you're taking a drug every day, you don't want to be high.
Magda does.
Good on you, Magda. On the positive side, when you take, I haven't taken MDMA myself, but apparently when you take MDMA orally, it gets taken up, it gets into the brain. It has great pharmacokinetic properties, and this is in part due to the amphetamine scaffold highlighted in blue here, which is the privileged structure in medicinal chemistry. It's got a fascinating polypharmacology, which means there are opportunities to tweak which receptors you're hitting in the brain for multiple different indications. It's got an excellent safety record. Millions and millions of people around the world have taken MDMA, and there are really very few adverse events, serious adverse events, that can be soundly attributed to MDMA. And it's got that demonstrated efficacy in PTSD psychotherapy. The question is, what else could it potentially be used for? So how does MDMA work? Well, as I said, alluded to, it's complicated. It hits lots of target in the brain. And its main effects are thought to be elicited through the increase in extracellular levels of these neurotransmitters, serotonin, dopamine, and noradrenaline. These are called the monoamine neurotransmitters. So this is a cartoon of two nerve cells. This is the presynaptic neuron and the postsynaptic neuron. The electrical signal comes down this neuron, and that induces a vesicle filled with neurotransmitters to bud with the cell membrane, releasing neurotransmitters into the synapse. And they move across the synapse and bind to receptors on the postsynaptic neuron, and that causes downstream signaling. This is how messages are passed between neurons in the brain. So that doesn't occur indefinitely. There are proteins that sit in the cell membrane called transporter proteins that bind to and pump those neurotransmitters back into the neuron, where it's repackaged in a vesicle for a new round of neurochemical signaling. So transporters for serotonin, dopamine, and noradrenaline, they're called SERT, NET, and DAT, respectively. Now what MDMA does is it binds to these transporters and is actually also a substrate for the transporter, so it gets pumped into the neuron. But in a kind of exchange process, that means that the neurotransmitter is pumped out. So in effect, binding of MDMA to these transporters reverses the flow of those transporters, increasing extracellular levels of the neurotransmitters. Now very crudely, serotonin, reversal of the SERT leads to serotonin release, and we believe that this is responsible for the pro-social and fear-extinguishing effects that make MDMA so useful in psychotherapy. Reversal of the NET leads to noradrenaline release, and that's responsible for the stimulant activity I mentioned. And reversal of the DAT releases dopamine, and this causes the euphoria and the reward that goes along with MDMA. So you see, we are mainly interested in compounds that retain this ability to release serotonin, and we're not so interested in releasing dopamine and noradrenaline. So we try and achieve that by the synthesis of analogues of MDMA that have similar structure but slightly different properties. I know there are some synthetic chemists in the audience, I'm glad of that, so someone will understand what I'm talking about here. How do we make these analogues? Well, we started off with racemic syntheses. MDMA on the street is always racemic, and in the clinic it's also being administered as a racemic drug, so a mixture of two enantiomers. And we make racemic drug by just doing a reductive emanation on these types of ketones. That works pretty well. We've investigated a number of different ways to make these piperonal ketones, starting off with a classical synthesis that's used to make many amphetamines. Start with piperonal and nitroalkane and subject those to cannabinoidal condensation. That gives a bit of nitrostyrene. And then reductive hydrolysis takes you through to the necessary ketone. It's a bit hard to get your hands on nitroalkanes of this type, and also this reaction doesn't work well when these substituents get a bit bulkier. So a more general synthesis involves starting with this piperonal chloride. You make a Grignard reagent, transmetallate to an organocuprate, and then add that to your acid chloride to give the corresponding ketone. Now, as you might imagine, that's quite technically demanding, not very scalable. So more recently we've gone to a more classical synthesis where we make an ester enolate from this readily available ester with an acid chloride. That gives us a beta-keto ester, and then we subject that to a Krapcho decarboxylation to give the ketone. And actually, just a couple of months ago, we've started exploring alpha arylation reactions. This is chemistry invented by Buchwald, and this works pretty well, and you can see this is probably the most efficient route, so we'll be picking up that in the coming period. Where a drug is chiral, it's usually preferred that a single enantiomer is marketed, so we have started targeting single enantiomers, and on this slide it's telling you how we do that. So we made both enantiomers of MDMA. That was a chiral pool synthesis. So these N-tosyl aziridines are available in a couple of steps from L-alanine, the amino acid. We take this aryl bromide, make the Grignard, and with copper catalysis you get regioselective ring opening disposition, giving sulfonamide. This can be N-methylated, and then the tosyl group can be removed with magnesium to provide, in this case, S-MDMA. And if you want the other enantiomer of MDMA, you start with the other chiral aziridine, which is derived from D-alanine. More generally, we use chiral auxiliaries to access single enantiomers. So, for example, we do a reductive amination with this ketone with alpha-methylbenzylamine, a single enantiomer. That gives a mixture of diastereomers now, which can be separated using normal column chromatography. Usually, often it's a difficult separation, but usually they can be separated. Once separated, we can N-methylate. Chiral auxiliary can be cleaved off through hydrogenolysis, and that provides us with the free base. And then we typically convert the free bases to hydrochlorides because they're crystalline. And that's important when we use this kind of approach because unlike the approach up here where we know which enantiomer we're making, down here we only know once we get a crystal structure. And that's a model based on crystal structure of the hydrochloride. We've also started using Elman's sulfinamide as a chiral auxiliary. So in this case, the reductive amination is done stepwise. Condensation gives this N-sulfonyl imine, and then reduction with sodium borohydride provides, again, a mixture of diastereomers. Where this R group is quite small, we usually get good diastereo selectivity here, above 90% of the major diastereomer. And then again, we N-methylate, and in this case, the chiral auxiliary is cleaved off with acid, which takes us directly through to the salt again. Okay, so with compounds in hand, how do we test them to work out what's useful to try and progress towards the clinic? This is sometimes referred to as a triage funnel. You want to put a lot of compounds in the top where your assays are not too expensive. They're all pretty expensive, I can tell you, but most compounds go at the top where the assays are not too expensive, and then as you funnel down, usually getting towards animal models at the bottom, they get more and more expensive, but you're testing fewer and fewer compounds. So we start off with target affinity for those transporters that I mentioned earlier, the DAP, NET and CERT. This is done with urethins, not the urethins based in Western Australia, but at various sites around the world. At the same time, we do an anti-target screen. So HERG is it's the gene that codes for, it's kind of complicated, the gene that codes for a component of a protein that is an ion channel in your heart. And if there are drugs that bind to that ion channel and inactivate it, if they're antagonists, it can lead to heart failure. And again, several drugs are being taken off the market because they increase the risk of a heart attack. So we don't want activity at HERG. I mentioned before 5-HT2B, we don't want agonists at that receptor. Also, any drug that releases serotonin, that is also a monoamine oxidase inhibitor. Monoamine oxidase is an enzyme that breaks down serotonin. So if you've got a drug that releases serotonin and it's an MAO inhibitor, it leads to dangerously high levels of serotonin syndrome, and that can be fatal. Once we've gone through those initial screens, we collaborate with the National Institutes on Drug Abuse to look at transporter functionality. So I mentioned before that MDMA binds to those transporters in neurons and reverses their action. There are other drugs that just bind to them and block their action. So selective serotonin reuptake inhibitors, some of you may have heard of. These are common antidepressants that just bind to the serotonin transporter and block it, but don't reverse it. So we need to know the functionality. And compounds that look promising we take into pharmacokinetic studies, and we do this with the Centre for Drug Candidate Optimization based at Monash University. And if a compound has all those suitable properties and suitable pharmacokinetic properties, we might take it into an animal behavioural model. Atuka do Parkinson's disease studies, the Lambert Institute do prosocial models, and epilepsy. So the idea is we funnel compounds through the assay, we develop structure-activity relationships, and we use that iteratively to redesign the next round of drugs that go through the funnel again. Ideally, we want to be honing in on drug leads. So here is a status update. I started this research back in 2003, but we started the Emiria collaboration in late 2021. We now have over 200 MDMA analogues in hand. About half of those are covered by our first patent, which was published in 2003. And all have been screened for monoamine transport affinity that I mentioned. The vast majority have no concerning affinity for those anti-targets. And as I said, we're focusing at the moment now on single enantiomers. And we have several disease indications in mind. Some of them are ongoing. One of them we've ceased because that didn't look very promising. And this is our drug discovery pipeline. I won't call it a development pipeline yet, we're still in the discovery phase. The idea is that we're going to, the in vitro and in vivo structure-activity relationships that we're developing will help validate modes of action for those different disease indications and allow us to establish robust lead optimization pipelines. Hopefully that can be done over the next 12 months. Now, hopefully you're still with me, not too hungry, and some of you are asking the question, Parkinson's disease, what has that got to do with MDMA? To explain that to you, I need to give you a background on Parkinson's disease. It's a complex neurodegenerative disorder that affects the entire body, but symptoms usually only manifest when this part of the brain called the substantia nigra, literally black stuff, dies off to an extent where symptoms arise. This part of the brain is dopaminergic. It produces dopamine, and there are neurons that have receptors for dopamine in it. And for unknown reasons, it dies in Parkinson's disease patients. This means that Parkinson's patients are deficient in dopamine, and being deficient in dopamine, they have a great difficulty moving because that dopamine, those dopaminergic circuits in the brain are required for the coordination of smooth movement. So the video that you saw here is of a young Parkinson's patient demonstrating a cardinal symptom of Parkinson's disease, which is bradykinesia, slow, stiff movement. Parkinson's disease is typically treated, therefore, by dopamine replacement therapy, and this comes in the form of dopamine precursor, L-DOPA. This gets into the brain and is acted on by dopa-decarboxylase, an enzyme, which restores levels of the neurotransmitter and restores movement. And L-DOPA really is a wonder drug. It allows Parkinson's patients to get their movement back, but again, it's not perfect. And inevitably, unfortunately, over a few years, serious motor complications develop. One of these is called wearing off. That's where the L-DOPA doesn't work as effectively or for as long. So patients spend more time in that kind of Parkinsonian stupor that you saw on the previous slide. The other more visibly disconcerting side effect is known as levodopa-induced dyskinesia, or L-DOPA-induced dyskinesia, LID. And this is involuntary movement that ranges from kind of embarrassing and annoying head wobbles, dangerous writhing of the body, and it's extreme, it's completely debilitating. And for this poor gentleman, he would be asking the question, is there any point in me taking my medication anymore? What has that got to do with MDMA? Well, this is where it comes together. This is footage taken from a BBC documentary that I saw back in 2001 from the BBC Horizon series called Ecstasy and Agony. And it talks about the story of Tim Lawrence, who's, again, a young onset Parkinson's patient. And you can see here he has quite severe L-DOPA-induced dyskinesia. Tim lives in London, and he found that when he took ecstasy, his dyskinesia was almost completely alleviated. Tim was a former stuntman, and you can see there he's quite adept at doing a dive roll. So Tim got in touch with some neuroscientists in the UK, one of those being John Brochi, and they set up a little clinical trial, N=1, where Tim was given on day 1 L-DOPA plus placebo, just a sugar pill, and on day 2 ecstasy plus L-DOPA. So his normal Parkinson's medication plus either placebo or ecstasy. On day 1 here, you can see this is before the L-DOPA's kicked in, so he's in that raw Parkinsonian state and unable to move, slight tremor, general bradykinetic symptoms. On day 2, for some reason, the L-DOPA has kicked in faster. That could just be random, but we don't know whether it's got something to do with the ecstasy. But you can see you saw before, he was moving quite smoothly. When the L-DOPA has kicked in on day 1, Tim was demonstrating that dyskinesia that we saw earlier, whereas on day 2, with ecstasy on board, he's moving completely normally. You would not spot him as being a Parkinson's patient. Now, that is obviously anecdotal research, but there are studies that have been done in various animal models that back up this in a statistically significant way. The other thing that this study showed is that ecstasy is able to extend the therapeutic duration of L-DOPA. In Parkinson's patients, well, first of all, you can see after 135 minutes, Tim is back in that Parkinsonian bradykinetic state. He can hardly move at all. That's on day 1 with placebo on board. On day 2, at the same time point, he's moving perfectly normally. So MDMA is able to extend that therapeutic duration. That's the time when L-DOPA works, where Parkinson's patients have their movement back, and that is in Parkinson's communities called on time. So before I go on, I saw this documentary as a postdoc. I was very interested in Parkinson's disease at the time, and I got in touch with John Brochi and we started collaborating. One of the first questions we asked is, what are the enantiomers of MDMA doing? As I said, it's a chiral compound. The MDMA that Tim Lawrence took was racemic. And with John Brochi's help, we found out that the two enantiomers act synergistically to provide dual benefits in Parkinson's disease. So R-MDMA reduces levodopa-induced dyskinesia, and S-MDMA extends on time. This is quite unusual. Most of the time when you've got a chiral drug, one enantiomer will have some useful activity and the other will just do nothing. It's the most common outcome. That didn't solve the problem of the psychoactivity of MDMA though. Tim Lawrence himself said, I don't want to be high all the time. So if we're going to use this for a treatment for Parkinson's disease, we need to get rid of that psychoactivity, amongst other things. So we asked the question, can the beneficial effects of MDMA in Parkinson's disease be dissociated from the psychoactivity? Now, I don't have time to go into the details, but the answer is, in animal models at least, yes. So some time ago now, we described the discovery of UWA-101, a simple analogue of MDMA where the alpha-methyl group has been replaced by a cyclopropyl group. This compound was shown to be non-psychoactive based on studies in rats, and it improves the duration of quality on time, that is, on time that's not affected by that disabling dyskinesia, more than any clinically available drug. There was a problem with 101, however. In the primate model that it was tested in at the highest level, it did seem to induce some psychosis-like behaviours. Dopaminergic drugs is one of the problems with dopaminergic drugs in humans. It's also seen in animal models, they do induce psychosis over time. But fortunately, one of the enantiomers of UW101, we looked at both of them. One of the enantiomers was completely inactive. The other retained its ability to the benefits in Parkinson's disease, but didn't cause the psychosis-like behaviours. So in this gold standard model of Parkinson's disease with dyskinesia, the dyskinetic MPTP lesion primate, 121 extends on time by 40%, increases good quality on time by 215%, and doesn't exacerbate those psychosis-like behaviours, as I mentioned. But we couldn't push this towards the clinic because this compound had been disclosed, so it couldn't be patented. The mode of action was uncertain. That's another thing that pharma don't like, and so more research was required. I can't talk about a lot of stuff, sorry, because it's the intellectual property issues. But these are some disclosable results. All these compounds are covered in our first patent. So this is taking us to the next level of testing where we're more closely examining the mode of action of UWA-121 and related compounds. This is done through collaboration with Michael Bauman at the National Institutes of Drug Abuse in the USA. And Michael is an expert in examining the functionality of those transporters I mentioned earlier, whether compounds reverse their flow and pump neurotransmitter out of neurons and into synapse, or whether they're just blockers of the transporters. So starting with the positive control, MDMA, you can see it releases, it reverses the serotonin, dopamine and noradrenaline transporters. These are EC50s in nanomolar, so the dose required to achieve half the maximal efficacy, and because it's an EC50, the lower the number, the more potent the drug is. Cocaine is the other control down here. In the model that Michael Bauman uses, which is rat brain synaptosomes, cocaine is a blocker of the transporters. So UWA121, we discovered, is a selective serotonin releasing agent. MDMA releases all three neurotransmitters. UWA121 releases just serotonin. It does bind to the dopamine and noradrenaline transporters, but it blocks them rather than causing reversal and release of the neurotransmitter. The enantiomer 122 also is a releaser, but significantly less potent. Change from a propyl group to a cyclobutyl group, slight increase in size, results in a loss of that releasing activity. So that compound now becomes a blocker, it's not a releaser. Extension of the N-alpha group to an ethyl group gives us a potency boost. So this is now about 5 times more potent, 4 times perhaps. Shares a similar profile, however, with 121. You can see also that the potency of binding affinity at the dopamine and noradrenaline transporters is increased, so this is a more potent blocker of the dopamine transporter and the noradrenaline transporter. So that compound was quite promising. We had a potency boost, it had a similar profile to 121. We wanted to take it into animal models, but before we do that, we need to test that the compound has pharmacokinetic properties. It means it's going to be around for long enough in the animal to actually have an effect. Unfortunately, based on an in vitro assay rat with the microsomes, it had a half-life of just under 3 minutes, which means there's no point putting it into a rat. It would just be chopped up too quickly and not have a chance to get into the rat brain. So a bit more on metabolism of these types of compounds. These are stabilities in liver microsomes, which is a liver preparation that contain metabolic enzymes, half-life in minutes. It's typical that drugs are metabolized more rapidly in rat liver microsomes. They just have a faster metabolism than humans. And to put this in context, so this is 47 minutes to 76 minutes. When a human takes 100 to 120 milligrams of MDMA, they feel the subjective effects for 4 to 6 hours. You can see as we go to 121 and then to 154, so N-methyl through to N-ethyl, there's that rapid drop-off in stability, in particular in the rat liver microsomes. And you might think, well, maybe that's because the N-ethyl group has been cleaved off more easily than N-methyl. But I can tell you that is not the case because we provided this primary amine to the CDCO who did these studies, and they told us that this was not formed, this is not a metabolite of these compounds, at least in this assay. There is a hint of what's going on here. The benzodioxyl moiety, this part that I'm circling here, in MDMA, is known to irreversibly inhibit cytochrome P450, particular isotype, CYP2D6. These enzymes are in your liver and they're important for oxidative metabolism of drugs. And when you get a kinetics plot like this, so this is drug reducing in concentration, and it suddenly plateaus off, it's a clear indication that the main metabolic enzyme is being inhibited. So it's a kind of suicide mechanism as the CYP starts to metabolize UWA154, for example, it gets inhibited and the metabolism slows down. Now that is known for MDMA and it's acceptable for psychotherapy because you have 1 or 2 drug sessions and then you don't take the drug anymore. But for Parkinson's disease, where the patient would probably be taking the medication every day, that's a problem. Because drugs that inhibit cytochrome P450s can induce drug-drug interactions. We need these cytochrome P450s to protect us from all the xenobiotics that we get from pollutants and drugs in our diet and so on. All right, so to conclude, the Parkinson's disease lead, UWA121, is a selective serotonin releasing agent. It's also a dopamine and noradrenaline reuptake inhibitor. Our working hypothesis is that DAP blockade of the dopamine transporter, is responsible for extending therapeutic duration or on time, and serotonin release ameliorates dyskinesia, maybe by counteracting the negative effects of dopamine, long-term dopamine therapy, dopamine replacement therapy, sorry. UWA154 is a more potent compound with similar functionality, but with both of these compounds, really, a longer half-life is necessary to have any real use in Parkinson's disease. 154 is a selective serotonin releasing agent with reduced half-life. And I mentioned earlier that that's one of our goals for psychotherapy. It's exactly what we want for psychotherapy. But at the moment, the half-life is too short for us to be able to demonstrate useful activity in animal models. So we need a bit more stability to be able to demonstrate the efficacy before we can think about going into clinical trials. So efforts to improve metabolic stability and avoid CYP inhibition are ongoing. And I'd just like to finish up by saying there are other SSRIs on the market. One of those is fenfluramine, and it's used to treat Dravet's and Lennox-Gastaut syndromes. These are devastating childhood epilepsies. You might have seen these on the news, for example, also treated with cannabidiol, but the treatments are really not very good. And that's exemplified by fenfluramine because it and its major metabolite, norfenfluramine, are potent 5-HT2B agonists. You might recall earlier I said drugs with this property cause heart valve recoverylopathy. So these poor kids are getting a drug that's probably damaging their heart, but because they've got such severe symptoms, it's kind of worth it. So we're just starting to look at some of our compounds in animal models with Ian McGregor at the Lambert Institute at the University of Sydney, epilepsy models. It remains me to thank the team of wonderful people I've been lucky enough to work with over the last few decades. In particular, my current group members, Adrian, Glenn, Jasmine, Adam and Mark. Former group members who kicked things off with the MUA Analogues program. All my collaborators, mainly biological collaborators. And the wonderful team from Ameria, Wayne, Tatiana and CEO Michael Winlow. They're more than just providers of funding, although that is much appreciated, but they really got behind the project. They're encouraging and have helped establish collaborations internationally, which have been really useful. I want to thank the West Australian Government for funding therapeutic innovation in Australia. Finally, I'd like to wind up by thanking my dad, who gave me the inspiration to work on Parkinson's disease in the first place, and probably to become a medicinal chemist, if I'm going to be honest. Dad lived with Parkinson's disease for nearly 30 years.
Right, let's thank Matt for his fantastic talk.